Hello Chem 213 students, and welcome back for our last video this week on peptides and proteins. When we left off, we were talking about how you can cleave a protein using enzymes and separate it into fragments, and then use the so-called Edmond degradation to determine each of the amino acids in the protein in sequence, and then these pieces can be fit back together in a sense to determine the entire sequence of the peptide. So we've talked about how we can determine the sequence of amino acids in a peptide, and so once these are determined, we will know how to produce a protein by producing that sequence of amino acids. And so we can synthesize peptides by producing that sequence of amino acids and creating that peptide or protein. So to form a peptide bond, the N-terminus of one molecule will be joined to the C-terminus of another molecule. And when this happens, it will be a condensation reaction, and we will lose one hydrogen and the water here. So I'm just going to say one hydrogen in the hydroxide, and this will come out as water. And then you're going to have a bond that forms between this carbon and this nitrogen. So in order to do this, we have a variety of coupling agents that can be used. DCC, dicyclohexyl carbodiamide, can be used, and you can see that pictured here, the structure. So what is the mechanism for performing a amide coupling using DCC? Well, we have nitrogen here with 3 bonds and 1 lone pair. This one also has a lone pair, although it's not shown. And this is going to undergo a proton transfer. And so, of course, the carboxylic acid's far more acidic than any amine. In fact, amines are, you could consider them more basic, the protonated form having only a pK of around 10 or so. 10 would depends, but we've seen in the past 10-ish to higher in some cases. And so first step here is going to be a proton transfer from the more acidic carboxylic acid to produce the protonated amine, which will be less acidic with a higher pKa. And once this occurs, the nitrogen will be making 4 bonds and have no lone pairs, and thus it will have a positive formal charge. The oxygen has an additional lone pair remaining after the proton transfer, giving it 1 bond and 3 lone pairs for a -1 formal charge. Now this carbon is attached to 2 quite electronegative nitrogen atoms, and so those nitrogen atoms are pulling electron density away from the carbon. In addition, we have this resonance arrow that can be drawn, and I'm going to draw that resonance arrow first here. So this resonance arrow, I'm going to switch over to blue for this purpose too, just to emphasize. And so in that case, the positive formal charge would lie on the carbon, having 3 bonds and no lone pairs. And so we can see a little bit more clearly after this resonance, but it's done all in one step here, as is often done in these kinds of cases in many of the reactions. So we've got the nucleophilic attack on the carbon combined with a resonance arrow here showing the pi electrons going on the nitrogen. This is kind of a terrible arrow because it's so small, but at that point, we have more of the amine, and so the amino group on another unit of this dicyclohexyl carboimide here is going to go ahead and perform a nucleophilic attack. Again, another resonance arrow here. This resonance arrow showing a pi bond between 2 atoms of differing electronegativity. This makes this a carbocation on this carbon, very attractive to this lone pair on the nitrogen being a nucleophile. So we get a nucleophilic attack, and now this nitrogen is forming 2 bonds to carbon and 2 bonds to hydrogen, giving it a +1 formal charge. This oxygen here now with 3 pairs and 1 bond, giving it a -1 formal charge. At this point, we have the loss of this amide as a leaving group here, and so simultaneously we're going to have 2 resonance arrows, one showing the reformation of the carbonyl bond, recovering that stability. Also, we have the resonance arrow here of the lone pair going back to the nitrogen. And finally, what's really happening is that this whole amide is leaving as a leaving group. And so it's no longer going to have a bond here to this carbon. And so now this we have this bond here between oxygen and carbon, a double bond. This oxygen here, this is no longer attached. There's a double bond here to this oxygen. So this is a byproduct molecule. And finally, this carbon has four bonds, 2 to oxygen with the double bond, 1 to carbon, and 1 to nitrogen. So that's this bond right here to the nitrogen. And then the remainder of the amine here. So the amine group can be the DCC. It can be something else. More likely, it is something else because that would form for us an alternative product. So you add the amine that you're looking for. There you have that carbon, and then you have the alpha carbon right here that nitrogen is attached to. And so you can carry forward a sequence. So this amine might be a larger portion of, so this would contain the amino acid with the side group, and this would be the larger portion of the peptide that had already had the other pieces attached to it. And finally, you get the amide bond, and this would, if you have a series of amide bonds, a series of peptide bonds, you'd get a whole polypeptide. So the coupling of 2 amino acids will potentially yield 4 different peptides, and this is because when you react this with DCC, you initiate with this with DCC, and then you have the attack of the amino acid afterwards. Now we have to control the way in which these add together, right? Because we can have the first amino acid here with the first R group, which we're calling R1, have the N terminus, and the amino acid with the R2 group have the C terminus here. Or we could have the opposite, the second, I'm just going to call this number 2 here, amino acid number 2 versus amino acid number 1. You could have amino acid number 2 have the N terminus, and amino acid number 1 have the C terminus, or you could have a reaction between the same amino acids. So the same amino acid here twice, having both the C terminus and the N terminus. So in order to control which peptide that we get, there are going to have to be protecting groups installed between each peptide bond formation to make sure that we get the nucleophilic attack from the nitrogen that we're interested in adding onto the chain, and not another one. So what we'll do is if we want to make a dipeptide where the N terminus is R1, peptide is amino acid 1 rather, and the C terminus is amino acid 2. What we're going to want to do is install a protecting group on the amine side of amino acid 1, so that it will not be involved in adding something over here on the N terminus side, so that will be the N terminus. And then we'll need to install a protecting group on amino acid number 2 so that its carbonyl is not attacked and we don't add the amino acid on the wrong end here, making it the C terminus. So we'll install these protecting groups between them. Then we react with DCC to initiate that reaction, and we can form the exact peptide bond that we're looking for, where the first amino acid is on the N terminus, the second amino acid is on the C terminus, and then we can remove the protecting groups and finally get the dipeptide that we're looking for. Again, the first amino acid here on the N terminus and the second here on the C terminus. So what is the nature of these protecting groups? The amino group can be protected as a carbamate, and so we react here with this carbamate, which has a very nucleophilic center here, which can be attacked. So again, we have the resonance arrow here, which produces a negative formal charge on the oxygen, a positive formal charge on the carbon, having 3 bonds and no lone pairs, and this will be vulnerable to a nucleophilic attack by the nitrogen here. And so then that nitrogen here will be making 4 bonds. There will be two to hydrogen here and 2 to carbon. And again, this oxygen here now has a -1 formal charge, having 3 bonds and no lone pairs. But this carbonyl will be reformed, just like reactions we've seen before, and we'll have a simultaneous loss of the leaving group. Whoops, not highlighter, pen here. We'll have a simultaneous loss of leaving group as that carbonyl is reformed right here. And so we'll lose the remainder of the carbamide, but now we have installed a protecting group on the N-terminus side. So we can see the nature of that protecting group. It is an ester group, so we can see here we're going to get a double bond, and on the same carbon we're going to have an oxygen. So we're going to have an ester group here, and so we turn this to this ester group. Again, we have the carbonyl here with the same carbon attached to oxygen, also attached to nitrogen. So in terms of what is commonly used for carbamate protecting group, ditert-butyl dicarbonate is generally used. So ditert-butyl and then dicarbonate is this central area here. And this will form for us a tert-butyl ether type group. It's kind of like or ester type group. It's kind of like an ester and an amide at the same time here. But we have this protecting group. So now the N-terminus side cannot undergo it will not be a good nucleophile, because this is going to be heavily electron withdrawing from the nitrogen, and this will not behave as a good nucleophile. Also, there will be steric hindrance, and so this is really going to slow down any formation, any nucleophilic attack by this nitrogen here. So again, here's the whole mechanism here. We have a nucleophilic attack and a simultaneous resonance arrow here, so that the carbon does not have 5 bonds. Then we get the loss of the remainder of the carbamide as a leaving group here, and that leaves us right here with a kind of ester type group right here, but also attached to the nitrogen. At that point, we have we have this oxygen here. This oxygen now is going to have 3 lone pairs and only 1 bond remaining. So that's this remaining piece here. And this will be not tremendously basic, but enough so that it can grab this proton to some extent from this protonated nitrogen, and then that will cause the nitrogen to then be neutral instead of positive. Here it has 4 bonds and no lone pairs. There's a positive formal charge, and here at the end it's a neutral amine or amide, I suppose, right here with this group. So the protecting the amino group of the amino acid, ultimately at the end, when you're done with your synthesis, you want to remove the protecting group. So this can be removed by a very strong acid such as trifluoroacetic acid, which will then behave to hydrolyze this and hydrolyze this this amide and ultimately give you back the amine. So the mechanism for that is it starts with the protonation of the carbonyl. So the protonation of the carbonyl is going to make it a better electrophile. And so we're going to have a proton transfer from the very acidic trifluoroacetic acid. At that point, we're going to have the breaking of the tert-butyl here to produce for us now this double bond here. Now we can't have 5 bonds between the carbon and the oxygen, so we have this pi bond adjacent to a carbocation, and so that's going to break this bond here. It's going to leave a carbon double bond oxygen here, and then we're just going to have a standard OH group. So we're going to have the carboxylic acid remaining. And so this carboxylic acid is quite acidic, and so to some extent, even though the trifluoroacetic acid is more acidic, to some extent we can remove a proton, and this is going to be this reaction is going to be propelled forward by the subsequent reactions that occur. This arrow is pretty terrible. I'm going to redo it, but you can see the arrow right there. I'm just kind of tracing over it. We're going to put that lone pair right back on the oxygen. And so off to the side too, we also have a side product of isobutylene, which can be created to some extent by the deprotonation of the tert-butyl group that left here. Somewhat stable due to the fact that it has a positive tertiary carbocation, but this is a side product, isobutylene. The point here is, though, we have this deprotonated oxide This is going to have 3 bonds and no lone pairs, or 1 bond and 3 lone pairs rather, giving a -1 formal charge. Now at this point again, the trifluoroacetic acid is extremely acidic, and so what can happen here is we can get the decarboxylation of this amide. This is going to form here a double bond between this carbon and oxygen, while simultaneously this group is leaving a lone pair on the nitrogen here. And this leaves as carbon dioxide. And now we have a proton transfer here, where we get the proton back on the nitrogen. And so that gives us nitrogen, 1 lone pair here, 1 lone pair right here from the broken sigma bond here, and we have 1 additional hydrogen. So this nitrogen now has 2 hydrogens attached. It still has this bond to this 1 carbon, and it has a lone pair. So 3 bonds and 1 lone pair, no formal charge. And then under these acidic conditions, again, trifluoroacetic acid being a very strong acid, this reaction will finish as a protonated amine. And so we'll have the proton transfer here, and this is simply because the pH is so low in the presence of this trifluoroacetic acid that the amine is going to be a protonated amine here. And so we're going to have a nitrogen with 4 bonds and no lone pairs, giving it a positive formal charge. And so this is the deprotection here of the Bach, and we get our bare amine group for the N-terminus returned. So an alternative to protecting the amino group with the Bach is the use of this 9-fluorophenylmethylchloroformate. And so you don't need to remember that name or anything, but it's a similar type of thing. You get this kind of ester group or ester amide / group attached here, and then you can use the same type of trifluoroacetic acid to remove the group, right? And so alternatively, you can use basic conditions to remove it as well, which is preferable. And so here we have a proton that is attached to a carbon that will be a tertiary, resonantly stabilized tertiary carbocation. I mean, look at as soon as we get this carbocation, it's going to be carbanion rather. It's going to be super resonantly stabilized. So we have this proton transfer. So why does this proton transfer occur, leaving these 2 electrons here as a carbanion? It's because you're going to have resonance all the way around these rings. I'm going to draw some alternative resonance arrows here, but this resonance will go all the way around this ring, also all the way around this ring. And so you get super significant resonance here, stabilizing this carbanion significantly, making this proton significantly more acidic. At that point, you can have you're going to have the formation of the double bond here, a cleavage of this bond, producing a carbon-oxygen double bond here. Of course, this carbon can't have 5 bonds, so simultaneously we're going to have cleavage of this sigma bond here. So this is going to produce for us right here. This is going to leave as CO2, and then we're going to have this remaining product here with this double bond and CO2. And most importantly, lastly, we're going to get this lone pair of nitrogen here to deprotonate the formerly protonated amine here, cyclohexamine, and then we'll finally get this nitrogen having 1 lone pair from the electrons right here from the cleavage of the sigma bond, and this lone pair now becomes a bond to a second hydrogen atom instead. And both of these reactions are driven forward by the evolution of CO2 gas. So when this is removed by Le Chatelier's principle, that's going to drive this equilibrium forward by removing the product. So the equilibrium drives forward to replace the CO2 that is leaving as a gas. In terms of protecting the carboxyl group can be protected by forming an ester instead of the carboxyl group. So methyl and benzyl esters are most popular. So this R group will either be a benzyl group or a methyl group, generally. And this is catalyzed by acid. So hydrolysis will then be used to deprotect the ester. or to turn the ester back to a carboxylic acid. And so the protecting group here will often be the benzyl ester. So benzyl meaning, remember this, it's attached at this carbon, one carbon away from the benzene ring. Then if we react with either H2 with platinum catalyst or HBr and acetic acid, we can then hydrolyze that ester and recover for ourselves the carboxylic acid. So if you're preparing a dipeptide here, what you're going to want to do is, again, you're going to want to protect the C-terminus of one of your amino acids that you want to be the C-terminus of the dipeptide, and you would do this, and then you want to protect the N-terminus that you want to be the N-terminus side. So let's say we want alanine to be the N-terminus side. Well, to protect the N-terminus side, we react with this Boc, and that will protect the N-terminus side. For glycine, we want to protect the C-terminus side, so we react with an acid, catalyst, and methanol to produce a methyl ester. At that point, we can use DCC to produce the peptide bond for us between the alanine and the glycine, and then we will react with trifluoroacetic acid to deprotect and break the amide bond to the Boc group, and then we'll use sodium hydroxide and water to hydrolyze the ester on the C-terminus side, and then we get the dipeptide. So this method of protection, DCC coupling, deprotection, is not practical for forming larger peptides. Basically, you have side reactions that, and these side reactions compound, and so the more times that you do this, the more likely there are to be reactions that you did not intend, and thus you will produce peptides that you did not intend to. So for every amino acid added to the peptide, 3 reactions and 3 purifications would be necessary. So this just becomes also cumbersome to continuously protect the group, do a reaction, and then deprotect the group. So there's an alternative synthesis called the Merrifield synthesis, which is a superior method, and it reduces the number of steps. The peptide is going to be tethered to an insoluble polymer. So what we're doing is, at this point, we're just building off of one end instead of building off of both ends, because one end of the peptide is just completely blocked by being attached to an insoluble polymer. So because this is attached to a solid, we call this solid phase peptide synthesis, or SPPS. So for example, the C-terminus of an N-protected amino acid is attached to the surface of a polymer. So here's the polymer. It's polystyrene, and and we have some alkyl halide groups here. It's similar to polystyrene, but we have some alkyl halide groups here, and these this is a benzyl group, and so the benzyl group can be used to protect the C-terminus side. And so the carboxyl group here goes and reacts with that. We get the ester. And then, assuming that the amino acid is already protected with Boc, we don't have to worry about any reactions of the amine side. We only will deal with reactions of the carboxyl side. And so you can see we have a benzyl ester now produced on the carboxyl side, and then we have the bare amine. And so notice too, we can do many peptides at the same time too. For every time we have one bond here to the polymer, we have many peptides that can now be added to, and so we can produce these in bulk and in parallel to produce a lot of these peptides. And so after this, we now take the next amino acid, we react with DCC, and that will react, and that's protected on the amine side as well, so it's only going to react on the carboxyl side, and so we get the peptide bond here attached to the carboxyl side with the next R group, this one right here, sticking out, and then we can we go on ahead and we deprotect and we add on and we add on. And so here we can add on to only one side one by one, and we can do a bunch of these peptides in parallel. And so this is a much more efficient way of producing many peptides that that we're looking to produce here. Once a peptide is finished, an HF is used rather to finally cleave, hydrolyze this ester bond and the C-terminus side is released, and the whole peptide is released at that time. So the advantage to SPPS is really the ease of purification too. As I said, if you're doing this by the simplified method, protecting both sides and then using DCC, this is not. This is going to produce a lot of side products, and you're not going to have a very pure sample of your peptides that you were trying to make. So the process can now be completely automated, and so there are machines, commercial instruments that you can feed amino acids into and these reagents, and they will produce for you the peptide. And Merrifield won the Nobel Prize for this work in 1984. So this is a relatively new synthesis, right? When I was in school, this synthesis was just starting to be commercialized. There were just starting to be commercial peptide synthesizers that you could buy. And previous to that, they were only in labs. Finally, when it comes to protein structure, the protein structure is not just determined by the sequence of the amino acids. That's one element of of a peptide or protein structure. But when those amino acids come out of the ribosome in cells and are produced one by one, the sequence of amino acids, those amino acids fold in on themselves and produce a three-dimensional structure to the protein that is determined by the sequence of amino acids and their attractions to one another, both on the backbone of the peptide sequence and also between peptide sequences, and in their interaction with their solvent, such as water, which is usually the biological solvent. There are 4 levels of protein structure. The sequence of amino acids is simply the primary structure. And the interactions between the backbone of the amino acids is what's going to be the secondary structure. The interactions between the side groups of the amino acids, such as disulfide bonds that occur between cysteine residues, that is referred to as the tertiary structure. And then when multiple proteins come together to form a protein complex, they are said to have a quaternary structure. So let's take a look at these in sequence. What you have here is an example of human insulin, which is it has a sequence of amino acids. It has all 4 of these structures. So it has a sequence of amino acids. It has some secondary structure where these amino acids interact with each other, some tertiary structure where we have disulfide linkages and other interactions between the side chains, and finally we have a quaternary structure because we have 2 amino acid sequences which are joined together by these disulfide linkages to produce human insulin. So the primary structure is the amino acid sequence, and so we've got 2 primary structures here for the 2 peptides that are joined in the quaternary structure of the protein. And so if we wanted to specify the primary structure of this protein, we'd have to specify the amino acid sequence for both chain A here with 21 amino acids and chain B with 30 amino acids. And remember, the side matters, right? The order matters. This side here pictured as the N-terminus, and over on the right is the C-terminus amino acid. The secondary structure has to do with the interaction of the portions of the peptide bond sequence here with one another. It's the localized regions of those. So it's largely dictated by hydrogen bonding interactions along the peptide backbone. So the peptide backbone is a series of amide bonds here and alpha carbons that produce the peptide. And when you have an amine group here, this amine group has a nitrogen bonded to a hydrogen, so this can donate hydrogen bonds here. The oxygen here is bonded to a carbon, making it very negative. So you have these carboxyl parts of the molecule that are partially negative, and then we have the amino groups here that are partially positive. And so these can give hydrogen bonds, and these parts can accept hydrogen bonds. And so you get significant interaction of these along the sequence, and this is what causes the sequence of amino acids to fold in on itself in certain particular ways. There are two primary ways in which this occurs. These are called the alpha helix and the beta pleated sheet. And in each of these cases, you can see we have interactions here between the amine groups and the carboxylic acid groups. These attractions, these hydrogen bonds, are what drive the formation of this secondary structure in the protein right here. In addition, when you have the alpha helix here, so this is the beta pleated sheet, here's the alpha helix. Again, this alpha helix is driven, the formation of this alpha helix is driven by these hydrogen bonding interactions between these carboxyl groups and these amine groups here, where the blue is the amine with a bond to hydrogen here, and the red is the oxygen. So we have this hydrogen bonding that occurs. Whoops, I think I got the wrong one there. Here I meant to circle, get those circles correct here. So we're looking for the amine here with its bond to nitrogen and hydrogen. This is going to be doing a hydrogen bond with this carbonyl group here, and there's going to be some interaction here as well. And so you can see that's going to be kind of repeating over and over. You're going to get a lot of those interactions. And depending on the nature of the side chains and the nature of the amino acids themselves, in certain regions of the protein, you'll have beta pleated sheet secondary structure, and in other regions, you'll have alpha helix portions of the secondary structure. There is shorthand that is used to represent these. So if you've ever seen protein diagrams, a great game you can play for about protein folding is called Fold It. If you look for that, and Fold It, and in just protein research here, Fold It. You can Google search that. In these three-dimensional structures that try to represent 3D shapes of proteins, alpha helix are generally represented by this ribbon form, and then beta pleated sheets are represented by this arrow that shows the sequence of the amino acids, its direction, and representing the flat part of the beta pleated sheet. And again, these beta pleated sheets have hydrogen bonding interactions that can occur either in the parallel fashion, where the protein is simply looping around in on itself like this, or where they're going in the same sequence, so it can come and loop around on itself like this, and then have hydrogen bonding interactions while this chain is proceeding from N terminus to C terminus parallel in this way. So here is the shorthand representation for human insulin. So it has chain A has a couple of alpha helixes in its primary structure. There are no beta pleated sheets. Also, chain B has one alpha helix region from the glycine residue 8 to cysteine residue 19. And then these two chains interact with one another by quaternary, by tertiary and quaternary structure. And so these yellow portions here representing the disulfide linkages between cysteine residues. So the tertiary structure of the protein refers to the 3D shape of the peptide chain, which is driven again by interactions of the side chains, generally with the solvent. So remember that some of these side chains are polar and some are non-polar. So the ones that are polar are going to be on the exterior of the protein, because that's where the water is. And so they're going to be attracted to the water. And so the folding of this sequence of amino acids, it folds, like a ribbon would basically in on itself, but the side chains that are polar are going to be pointing towards the exterior because they're going to be attracted to water. And the non-polar side chains are going to be towards the interior of the protein because they will be attracted more to the other non-polar side chains than they are to water. Now, if you disrupt this 3D structure of the protein by, for example, increasing the temperature, which will cause the protein to jiggle, and this may overcome the forces of attraction between certain portions of the tertiary structure of the protein, and it will unfold. And once it unfolds, it usually doesn't fold back into place. And this can be done by temperature. It can also be done by reactions of the side chains with acids or bases, so by changing the pH, and by by other ways. For example, we're reacting with oxidants or otherwise reacting the proteins with other things. So usually when we want to denature a protein, we're doing it because we're cooking. When you cook meat, you're denaturing a protein, or cook an egg. And that's why the egg, for example, is no longer soluble in water anymore. The proteins in the egg, they become denatured, and when they unfold, the nonpolar portions of the molecule are exposed, causing it to be less soluble in water. We can also cook things with acid, though. If you've ever had ceviche, that is the cooking of usually seafood using acid rather than temperature, which also denatures the proteins and cooks it just as well, just without temperature, with pH instead. Finally, the quaternary structure of proteins results when multiple peptide chains aggregate to form a protein. And so an example of that was insulin, as you just saw, and hemoglobin is another one. It's got a quaternary structure of 4 different proteins that all interact with one another, and in the center is a molecule called heme, which contains iron in the center of each one of these. And that iron can make a covalent, or can make a bond to oxygen and O2, and then carry that oxygen around the bloodstream. And so that's an example of a dissolved protein in our blood, hemoglobin, with this quaternary structure. There's other proteins that make up our body, for example, our nails and our hair, that are not soluble in water. These are called fibrous proteins, and the reason why they have the rigidity that they do is because they're formed by bundling together several polypeptide chains that have long sequences of alpha helixes, and these and so in hair, these are called alpha keratin proteins, these fiber proteins, and they coil around one another. The helical structures themselves coil around one another and produce thicker strands here, and these are the strands of hair that you see. In addition, these alpha helix structures are held together by disulfide bonds between their cysteine residues. So as I mentioned, we can reduce these proteins to unbundle these chains from one another, and then we can form the hair into the shape that we want, and then we can oxidize the cysteines, the thiols again. And remember, when you have the 2 thiols on the cysteine, when you oxidize them, they don't form any kind of, they don't do like the ketones or the alcohols and form carbonyls. Instead, you get a loss of hydrogen and you get the disulfide linkages. And so this is going to be oxidation because we're removing hydrogen. And when we oxidize, we reform those disulfide linkages, and the hair stays in the formation that you wanted it to be in, that you were trying to chemically alter it to be in. Now, for the reactions of forming proteins, for example, that we did synthetically, these tend to have a significant activation energy. When we're doing a synthetic protein synthesis, it can be quite time consuming because these reactions will take much longer. But in the body, the formations of peptide bonds and other reactions that occur are sped up by proteins themselves that are called enzymes. So enzymes are special proteins that have a special shape that can shape and properties, chemical properties, that make them behave like catalysts, and they can reduce the activation energy of many biological reactions, and these lead to far faster reactions. So many of the metabolic reactions that occur in our body would not occur at nearly the same speed in a test tube. But with the help of particular proteins that are meant to be enzymes for these biological reactions and speed them up by lowering the activation energy, these reactions can occur millions or billions or even greater numbers of times faster because of their significantly reduced activation energy. Some proteins are used for transfer. We mentioned hemoglobin. In each of those 4 proteins in the quaternary structure, there's a molecule called heme, which contains in the center of it an iron atom. That is our primary reason for dietary requirement of iron. This iron can chemically bond to oxygen, O2, and then that it can carry that O2 around the bloodstream, where it is later released into the appropriate region of the body where oxygen is needed. So yeah, this is referred to as a prosthetic group. It's not a protein itself, but it's a cofactor along with the protein that will achieve a particular reaction, chemical property. And that's going to be it for proteins. So I hope that was interesting to you to learn about the way proteins can be synthesized. In the body, they're synthesized in similar ways, although they are directed, as I said, their synthesis is directed by enzymes, which significantly decrease the activation energy and increase the rate of formation of these proteins, and the rates of other reactions as well. And so that's the end for the protein chapter. Please let me know if you have any questions, and have a great week and day. Day and week. Have a good one. Bye-bye.